Um, Good morning, girls. Today I'm going to talk to you about cell division. So important to know that every cell is derived from pre-existing cells. So not a single cell actually appears from scratch or is put together from scratch. There always has to be a cell that then divides to make more. And cells are produced for growth. So when you're growing, it's mostly because you are growing more cells. It's not because each individual cell is becoming bigger. But you also need to replace damaged or old cells. Some of the tissues have quite a high turnover and get replaced all the time. We're also going to talk about cell division and how it differs in prokaryotes, which are bacteria, and you might remember that these were the cells that don't have a true nucleus, and the eukaryotes, which is all the other organisms like unicellular organisms like protists, fungi, plants, and animals. Why is it important to keep cells identical? As you know, the instructions for making cell parts are encoded in the DNA. So the DNA is basically telling the cell what kind of cell it is, which proteins it is supposed to make, and what kind of job it is doing. So each new cell must get a complete set of the DNA molecule, because otherwise it wouldn't be functional. To ensure that, DNA must be replicated before cell division. And as you now know, during cell replication, one strand of DNA uncoils, gets separated, and each side is used as a template to add another strand of DNA. So in the end, you have two identical copies of DNA. And one copy can then go into one of the new cells and the other copy goes into the other new cell. And this ensures that both cells get the identical and full genetic information they need to function properly. So as a result, mitosis gives you two identical daughter cells. So when you're asked for the purpose of mitosis, the keywords you need to say is mitosis makes two genetically identical cells. Genetically identical is important, so they get the exact genetic information because they get identical copies of the original DNA. So why do cells undergo mitosis? Obviously to grow, which is the formation of new cells. In animals, that involves growing from a zygote. A zygote is a fertilized gamete, so a zygote is like the fertilized egg. And this then grows into an adult organism, and so you grow from one cell, which was the zygote, to about 50 trillion cells in your adult body. Plants keep growing all their life because they continue to make new seeds. The seedlings need to grow. They continue to make new buds, flowers, leaves, and so on and so forth, whereas animals usually stop growing at a certain age. It's not true for every animal, like fish, for example, continue to grow, but most animals eventually reach their maximum size and start growing. However, you also need to, cell division keeps happening because damaged tissue needs to be repaired. For example, you are constantly shedding skin cells and they need to be replaced, and you're also constantly shedding the inner lining of your intestine and that needs to be replaced. Cells need to be replaced. For example, you constantly make new red blood cells, whereas other cells in your body turn over a lot more slowly or actually not at all. Like, for example, brain cells, And therefore, the rate of mitosis differs quite a lot between tissues, and it is also influenced by temperature. And the reason it is influenced by temperature is because temperature influences the activity of enzymes, and as you know, enzymes control everything. And then obviously the availability of nutrients. If there aren't any nutrients to build new cells, then it can't happen. And mutations, some cells that undergo mutations become cancer cells, and they keep growing and reproducing uncontrollably, even though no new cells are needed. The DNA in our body is organized into chromosomes, and that's what a chromosome looks like. So let's have a look. In a closer look, in a bacterium, we have only one chromosome. So a bacterium doesn't have that much DNA because it's not that complicated. And it's just one strand of DNA in a circular form. However, when you look at eukaryotic cells, they have actually quite a lot of chromosomes. And the reason is that they are a lot more complex, and therefore they have to store a lot more genetic information, and they have a lot longer chromosome. So we don't really know why we have chromosomes, but biologists believe that it is a way of how the cell organizes all its DNA. So instead of having just one tremendously long strand of DNA that could usually get tangled, easily get tangled, it is chopped up into nice little packages, and that's what we call a chromosome. So a chromosome is basically a chunk of DNA or a strand of DNA. So the DNA is very tightly coiled around little protein beads. No. Normally the DNA is uncoiled. And here is a picture that's been made with an electron micrograph, and there's a very high resolution, so you can actually see the uncoiled chromatin, and you see all these really thin strands of DNA in here. So this is the strand of DNA. And under normal microscope that just appears gray, and we can't see any chromosomes. And so this is called chromatin. However, before cell division, the DNA gets very tightly packed. First of all, it gets wrapped around protein beads, which are called histones, and then it gets up these histones, and this then coils up further and coils and coils and coils. We call that supercoiling, until we get quite compact structures, which we call chromosomes. So that's what it looks like when we look at it under a microscope. And before cell division, they appear as X's. And the reason they appear as X's is before cell division, the DNA replicated, and we know it needed to replicate to make sure that there is one strand of DNA for each of the daughter cells. And the replicated strands stay together. They are held together here by a structure called a centromere. So you can see one strand of DNA here, and the sister strand, the identical sister strand, here. And this is where they're held together. And we call these two things chromatids or sister chromatids. Now during cell division, this structure divides here. So the centromere breaks, and then one strand, one sister chromatid goes into one of the new daughter cells, and the other one goes into the other new daughter cell, and they become the chromosomes of the resulting cell. So this is what we see before cell division. Between cell divisions, the DNA is uncoiled, and it looks just like this gray chromatin that we saw in the on the slide before. So before cell division, when we take a picture of chromosomes, then this is what it looks like. And we call this thing a karyotype. So what they've done here, they have dyed the chromosomes, so different sections show up in different colors. And you can see this is where the centromere would be. So you can see that they usually have a long and a short arm. And just by looking at their shape, you can arrange them into pairs. And this is called a karyotype. And you see here that there are 22 pairs where the chromosomes are identical. These are the so-called autosomes. So chromosomes are called autosomes. And then there is one pair, this one down here, where the chromosomes are different. And these are the sex chromosomes which determine our sex. So XX would result in a male, and XX would result in a female. Here is an electron micrograph of these chromosomes, and you can see the DNA strands in here and how it is coiled up. So this would be an X chromosome, and this is a Y chromosome. Now a cell can reproduce asexually and just make two identical daughter cells. And mitosis and binary fission are examples of asexual reproduction. Mitosis happens in eukaryotic cells. These are the cells that have all these chromosomes, and then they need to go through all these phases. Binary fission is what happens in bacteria, where everything is a lot easier. And then there is sexual reproduction, where the cells make two different gametes, an egg and a sperm, and then they come together to form a zygote. In sexual reproduction, the resulting gametes are not identical to the original cell. Sexual reproduction is called meiosis, and that's what we're going to learn about later in the year. Now, something needs to tell the cell when to divide. So cells go through so-called cell cycle, and their division is carefully controlled. And that's really important, because otherwise you would constantly grow little lumps in random places on your body, and you obviously don't. So your cell knows exactly whether it is supposed to divide or not. And this is called the cell cycle. So I'm going to stop that video here and then come back later with another one on cell cycle.